our first Decoding Thursday conversation brought to you by Avrio Genetics. I am thrilled with this 6-part series on genetics, epigenetics, and fertility. We have a special guest joining me today. His name is Dr. Brad Wilson. He has a PhD in genetics from Howard. He's joining us momentarily. I'm going to add him to the Instagram. There he is. He is a fabulous colleague of mine. And Avrio means tomorrow in Greek. I love this company. I love this brand. They're looking to make tomorrow a better place for all of us. One of the things that they absolutely do. Hey, Brad, how are you?
Well, thank you.
Welcome to Instagram, man. We have an amazing community. They're very engaging. They're going to ask you a lot of great questions. But I do want to thank Avrio for sponsoring this. You know, we're so lucky that we have so many great people who are bringing free content to our community. And this topic of genetics is tough. You know, Avrio wants to make it possible that everyone who wants to have a child can fulfill their dream of becoming parents. And we have this 6-part series that you're kicking off on the topic of genetics. So we're going to kind of dive right in. I think that we have to start with a very top-down approach on kind of what is genetics. And you just published a blog on this. So you're so good at explaining how the immune system and genetics is involved in fertility. But why don't we kind of start there in the backdrop of like genetics and fertility itself?
Sure. So when you're talking about fertility in general, it's you have to kind of realize that it's really truly a miracle that we are all here because of so many things that has to happen and have to go properly for us to for us, you know, to create a new human being, right? With regards to producing eggs and sperm from the female and male respectively, they have to come together successfully, and the sperm has to fertilize the egg, and then that egg has to gets transported to the uterus, and it has to implant properly, and then the whole growth and development needs to take place normally. And so there are so many places along the process where things can go wrong. It's really a broad scope and a broad conversation when you talk about infertility and genetics, because there are genetic factors that can impact a myriad of things, from anatomical structures to ovarian function, sperm production, implantation, the lining of the uterus.
You're right. It's a miracle.
It is. It truly is. The miracle of childbirth. And so for us to make it here, a lot of things had to go right. And genetics plays a part in all of that biology. So at any point along the process, things can go wrong, and there are, again, genetic factors that play a role into all those things. So it's very broad. Let's
actually start with the genetics of the embryo itself, because I think one of the biggest misnomers out there is when people do genetic testing of embryos, they're assuming that their baby is not going to have any genetic issues.
Right. So that actually makes it's a good segue, a good point in terms of the different stages or different time periods at which you can get testing. So before you have children, you can get carrier screening. And then if you're, after you utilize IVF, then there may be some screening done at that point as well to rule out genetic issues that you would, want to avoid or eliminate. And so in terms of genetic testing for an embryo, there's usually an array of conditions that are tested for that would, kind of be, our main issues that you kind of want to rule out prior to implanting or doing an IVF procedure. So those types of that type of testing includes things for diseases that are that will that you can detect at that stage through genetic testing, through like chromosomal abnormalities, et cetera. And then after implantation, then you move on to a different set of testing. And then when you're born, there's a set of newborn screening, that's genetic testing, right? So there's testing at different stages in the process, and they test for different things.
Once you're born, that's your set of genetics.
Right. Once you're born, I mean, well, once your egg is fertilized, I mean, that's the basic set. But there are things that can happen in utero along the way that can cause some issues. Environmental factors that can come into play that can disrupt that biology of the development of the fetus. But
and we're not also doing whole genome sequencing of an embryo. So correct. You have all your genetics once it's fertilized. That is the same. Right. If they did whole genome sequencing of an embryo, it would be the same as doing whole genome sequencing on the infant, correct?
Correct. Now, barring some mutations that could take place during that time.
During development, right?
During development, exactly.
Yeah, so there are changes because the cells are continuing to divide and multiply and so forth and so on until you show up. But I think the important thing there is that when you do PGT, or you're only testing a few of the genes, as opposed to the whole genome. I mean, how big is the whole genome?
We're talking, trillions of base pairs. So it's
pretty big.
That's a lot.
If you laid it out, how long would it be? How many football fields?
You're getting me with the.
Or how many trips? How many trips to Mars and back?
I don't have that stuff honestly. But yes, the molecule, if it's stretched out end to end, it would stretch a very long distance. But remember that all that information is packed inside the microscopic nucleus of your cell.
Yeah.
Again, one of those amazing things of biology, Mother Nature.
Completely, and that your eye cell and your nose cell, right, or your skin cell, it all has the same. It all has that same trillions of pairs.
Correct. Except for a couple of barring a couple of cell types like red blood cells, they don't have a nucleus, so your genetic information isn't in your red blood cells, but your white blood cells do have nuclei, and it's found there.
It's amazing. It's absolutely amazing to me. Okay, so let's break it down a little bit deeper, even into kind of the PGT conversation. You're not a PGT geneticist, but understanding that we don't test for everything at that stage, and yet part of the reason that PGT exists. Let's talk about PGT-M that can rule out monogenetic diseases, although it's up for debate if there's really a monogenetic disease anymore, right? Because we're finding that there's polygenes that go into almost every disease nowadays. But PGT-M, you can rule out cystic fibrosis, you can rule out BRCA, so you can actually self-select which embryos aren't a carrier of a devastating childhood disease.
Correct. So, for example, cystic fibrosis is a large gene, and there are actually hundreds of mutations in that gene that can lead to cystic fibrosis. So that's actually dependent upon the test itself and what variants it tests for. So if you're utilizing a test that, and I'm not sure if there are any that cover all of the mutations in cystic fibrosis, but usually they will cover the most common mutations found, and to rule out those mutations and others of that type. And yes, there are some diseases that are monogenetic. Sickle cell disease, for example, it's a single gene, a single mutation in the beta globin gene.
Wow, single mutation even. Wow. Okay.
Right. That causes that. I'm a carrier. I have sickle cell trait, so I have one copy of the variant that causes sickle cell disease. But the interesting part about that is that one copy is protective against malaria. And that's why we often find it. That's why it survived, right? That's why it survived in our population, because it offers us a protective advantage. So I can't get malaria.
That's amazing. Oh my gosh. And when the world opens back up again, you should travel to Africa so that you can just put that piece of data to work.
Oh, definitely plan to go back at some point. I've been twice. I've been fortunate.
You have? Oh my gosh, that's amazing. Okay. I did not realize that, because I have two cousins who are also sickle cell trait carriers.
Yep. So
Yeah, and
they too are essentially immune from
from
malaria.
malaria. That's amazing. Okay, I didn't realize that. So I want to kind of keep going down this pathway of like, okay, so there's certain things that we test for. In the case of just PGT, with like when I did PGT, I just was looking for for a normal embryo, one that was, euploid as opposed to aneuploid, because I'd already had a number of pregnancy losses and I only had a small cohort of embryos to work with, and I couldn't make new embryos. So that really was, the reason for doing it. And so in that case, they're just looking for chromosomal normalcy, if you will, right?
Right.
And
Right.
so what are they really looking at? Because I knew from doing that particular test that I had boy embryos and no girl embryos.
Right. So they're looking at essentially a chromosomal analysis and looking for the correct number and looking for any anomalies or abnormalities in chromosome number,
Okay.
or potentially some translocations that can be picked up, meaning a piece of one chromosome is broken and then attached to another piece of chromosome, another chromosome.
Mm.
And when the chromosome during development, one of those translocations can result in an aneuploidy or an extra copy of a chromosome. Say if you have, for Down syndrome, for example, as a common example, a piece of chromosome 21 can cause Down syndrome from a translocation,
Okay.
one of the mechanisms that will give you an extra copy of a given chromosome.
Okay,
So,
got it.
yes, so that level of testing is looking at gross chromosomal abnormalities and looking for, again, the appropriate number. And that's why you could tell if you had boys, because you determine if has an X or a Y, that particular
Right.
embryo. Mm-hmm.
Okay. And normally in what we do know too, that the vast majority of miscarriages, especially ones that are post week 9, it's typically a chromosomal issue, trisomy, right? Or something that would prevent that fetus, that embryo that you had your hopes in, from ever even developing into a healthy child.
Right. Looking for, anomalies that we are aware of and have categorized, and that are that can be deleterious in terms of if they are present in a fetus. So considering the resources that you're expending to have the child, you wouldn't want to implant an embryo with those types of anomalies, because then you'd be certain to have a child that is affected with those conditions. So
Right.
We have the ability to select and choose, and so you can avoid those issues.
Well, one of our audience members, Mel, she had twins, and one had cystic hygroma, and unfortunately, that twin didn't make it.
Right.
Yeah.
That's an interesting. That brings up an interesting fact that even identical twins technically may not be genetically identical, and that there can be,
Interesting.
a few differences in variants or
Wow.
mutations present in one that are not in another, if they are, if they are quote unquote identical, meaning from the same and a single embryo that split or divides into two,
Isn't
as
that
opposed
fascinating?
to two eggs that get fertilized. Yes, it is. It was really cool. It was kind of a misnomer, a myth that that they're genetically identical too. I mean, by and large, they are more genetically similar than two unrelated individuals or even two siblings, but they still may not be 100% genetically identical.
That's amazing to me, but it makes sense because we've often talked about if you biopsy an embryo in a different place, that you might not have the same results. And so people talk about that a lot with mosaicism, where, we have kind of a blended result and a lot of debate around whether or not to transfer a mosaic embryo or not. And it's very kind of clinic specific if people are comfortable with it. Sometimes it's up to the patient, the intending parents, if they want to give it a go, depending on the cohort of embryos that you have. But that makes a ton of sense to me, that once it divides, there's a lot of genetic development that's happening. And at that point, like you're saying, there could be mutations and there could be things that shift and change. Oh my gosh, it's a fascinating science.
Absolutely. So every time that embryo cell divides, it has to replicate the DNA in the cell. So during that process of replication is where errors can occur. And then that an error that occurs in one cell in an embryo can be the event that creates a mosaic embryo, and that one cell that had a genetic mutation continues to divide and among the remaining of the cells in the embryo that do not have that mutation. So then you can find, you can have an occurrence where if you biopsy in one place versus another, you can get different results based on that mosaicism.
Right. Makes sense. Okay. Now, Lauren, who's successfully pregnant right now, she had a PGT embryo that was normal, but it ended in a blighted ovum. We also know that a small percentage of people have PGT embryos that were normal and also had a pregnancy loss. So it kind of goes to your point about how many things have to go right and why it feels like every single one of us is such a miracle. So what are maybe some of the genetic changes that occurred while the embryo was trying to implant that could still lead to a pregnancy loss or a blighted ovum?
Sure. And another thing to keep in mind is that although we do some testing, we still don't completely understand the entirety of human pathology, all of the things that can go wrong from the genetic level. So if we're not aware of the mechanisms or the genes involved in certain things, then we don't have a test for them yet. So so the things that were tested for during the PGT were normal, but clearly that's not the entirety of things that that could be tested for clearly, as there was still an issue with that particular pregnancy.
Okay, got it. Before we move on to what you can test genetically during pregnancy, I want to stop for a moment and just say thank you to Avrio for sponsoring this. We are so grateful that we get to bring free content constantly to our users. You can ask free questions on the Fertility Answers app. And this is a 6-week Decoding Thursday series, starting with Dr. Brad Wilson on covering some tough topics, genetics and epigenetics, so that Avrio can help to deliver a better tomorrow by making it possible for everyone to have children and fulfill their dreams of being parents. And it's so aligned with our mission to help 100 million healthy babies be born to healthy parents. And so much of that is genetic. So how much of the outcomes that we're looking at in life, Brad, are genetic versus lifestyle?
Well, that's a good question. That's a great question. So there are a good number of diseases, disorders, conditions that we understand something about the genetics of, that we can test that some, and a large majority of which can be tested. Some things we still unclear about the genetics, as you mentioned, those polygenetic disorders that we're still doing the research on in terms of these large research projects. But in terms of in adulthood, many of the chronic diseases and illnesses and conditions that affect us are well, it could be considered a good thing in that they are environmental. For example, we'll take the biggest three out of them in terms of diabetes, hypertension, and heart disease, cardiovascular disease. So diabetes, that's really connected to your consumption of high fructose corn syrup and products that contain that. In terms of your consumption of that, and that's probably most likely the largest driver of diabetes. And then similarly with hypertension, it's regarding your sodium intake. There's so much hidden sodium in foods, particularly canned foods, that's there that you're not even aware of, and that's can be a major driver, environmental driver of hypertension. And then cardiovascular disease, of course, it's also fat intake, and particularly what type of fat is you're consuming animal fat versus plant fat.
So everyone has risk of that just by ingesting too much of the wrong thing, everyone. But some people who are genetically predisposed to those conditions might have an onset that's earlier on in life if they're consuming a lot of those foods, right?
Right, exactly. Hypertension is a good example of that. As a result, actually, the transatlantic slave trade acted as a selective event, put a selective pressure on that population in terms of the journey across the Atlantic. You're traveling across the ocean. The ocean is safe. Has salt water, and we know what salt water does to us. It dries us out. It pulls out the water out of our bodies. So if you were not able to hold on to your salt tightly enough, you may not have survived that journey. And as a result, the descendants of people who did survive the journey maybe genetically more hold on to the salt more tightly. And so then you put them in an environment where there's tons of salt in their food and they're holding on to that salt so tightly, result sky high hypertension. And then even some cases where people eliminate all of the salt from their diet, and it's still called what's called essential hypertension, meaning we don't know what's causing this, but we know it's there.
Wow.
Even after all the salt has been removed from their diet.
Wow.
So that's most likely strong genetic factors driving something like essential hypertension, where you removed the environmental factor of the salt from the diet.
That's fascinating. Okay, so the good news is, everybody, what you put in your mouth is up to you. It's totally up to you, which means that you have the most profound influence over your genetic expression, right? So that's important, especially during pregnancy, right? So let's talk a little bit about, what we put in our mouth and the epigenetic changes that it could influence on. So first, I think we're gonna have to define epigenetics. Let's start there.
Sure. Epigenetics refers to essentially this cool system that works to turn genes on and off. And that's another thing that people don't realize. Your genes respond to your environment. So there are certain genes that get turned on under certain environmental conditions. For example, there's a set of genes that are called hypoxia inducible genes, meaning when your brain is under oxygenated, or your body's, if you're low on oxygen, these genes get turned on under those conditions.
So people who have maybe altitude sickness maybe are more sensitive to those genes, like maybe react a lot faster than somebody else's.
Altitude sickness is a good example where that set of genes there's a good chance that those genes are playing a role in that condition, because they are responding to that environment where that person is in a low oxygen environment and they get turned on. And so there are, there can be short-term changes and long-term changes. Some other long-term changes are actually in response to trauma that's shown in descendants of Holocaust survivors and things of that nature, where there are epigenetic marks that are heritable, that can be inherited from your parents, that are a result of trauma that they experienced, or even more distant relatives.
Interesting. And so when we're talking about the genetic changes that can happen in utero, right, we're saying you've got these set of genetics, but mutations can happen along the way as the baby's growing and all those cells are multiplying and dividing. So how does what mom puts into her mouth, which I know a lot of pressure on us when we do finally get pregnant after all of our work, and I'm the first to admit that my food aversions and my food cravings completely won out over any book I had read prior to getting pregnant. But what is that impact, right? Why do we say take folate, especially methylfolate these days, the activated form, if we didn't, and that can result in spina bifida and other, issues, but that's an epigenetic change, right? That would be an epigenetic change in the positive direction, where we want to prevent that from happening.
Right. So the folate, the relationship of folate and what we more largely called neural tube defects, which there are several different types. Well, the folate is one of those essential vitamins that are important for the development of the nervous system. So it's not necessarily an epigenetic change where you're turning on or off genes. It's just what's required for the normal development.
I see.
So there's another, and actually there's another way that epigenetics comes into play in terms of we have two sets of chromosomes, one from our mom and one from our dad, but we don't express all of the genes on both sets of chromosomes. Sometimes we turn a gene off on one of the chromosomes because we only need one copy to be turned on. So that's another way that epigenetics works in terms of turning on
interesting.
a copy from one parent or another, because you don't necessarily need both copies to be expressed at the same time.
Okay, so here's a question breakdown for everyone. And Rocky, I'll get to your question. I think it's a really good one. So my father came back, he's 60% Italian, but I'm only 9% Italian. Right. So back before genetics, we thought mom and dad 50-50, right? So if dad was 60% Italian, I'd be 30% Italian.
Right. The math doesn't quite work that way.
No, it doesn't.
There's this amazing thing called, genetic recombination. And so it's a random assortment. So in terms of then you have another whole parent that's involved in this too, right? So
yeah.
In terms of your mom, in terms of her ancestry, is most likely that she's probably, I guess you were assuming that she's Italian also, or?
She only came back like a teensy tiny bit Italian.
Right. So
yeah.
You're not going to get all of your gene.
It's like I got his 40%. It's like his 40% Irish, Northern European, and my mother is like 60% Irish. It's like those combined together and created me with like a sprinkling of the Italian in. Yeah, but it's so bizarre that I think we all kind of our minds blow when we do like Ancestry.com or 23andMe, and then you think about that with your own children, right? I have a donor conceived son, so he's more Italian than me. Right, so, and that was a big shock.
Right. So clearly the donor had a considerable amount of Italian ancestry. Yes. And that made its way into Dante.
and the donor at the time didn't know, because they weren't doing expanded carrier screening or kind of expanded genetics on donors 13 years ago when I selected the donor. So the donor had no idea that he was Italian.
Right. Yeah. Often ancestry is a mystery to folks in their own family.
Right, exactly. So Rocky's asking, we get a lot of questions about the MTHFR, gene, and it's something that the IVF community doesn't know a lot about, so they don't like to talk about it. And it's something that most prenatals are adding methylfolate into multivitamins now. So it's not so much that people even have to think much about it as it relates to the pregnancy itself and protecting against those neural tubule defects, because we're taking the activated form anyway. But is there any science about other considerations on the MTHFR mutation that could negatively impact either your ability to get pregnant or stay pregnant?
So MTHFR specifically, I don't want to say no, because and I can't say no because there's so much that we don't understand yet. There's so much that we haven't figured out because we haven't done the type of research that will, kind of unfold all these things and kind of uncover these things. And then we're talking about, in terms of reproductive medicine and biology, that this is one of the trickiest areas. It's like trying to study the brain, right?
Right.
It's one of those things that are tough to do. Well, for the brain, it's because we have to develop the right tools. But in terms of reproductive genetics and technology, it's really been the technology itself in terms of our ability to to extract information from the genome. And we just haven't done large enough studies yet to really tease out all the information in terms of all the things that affect us. But we're in but these large projects that are, being run by, government entities and agencies. That's the intent and the goal of those large projects, is to start to uncover more of the genetic causes of these diseases, like these polygenic diseases that you mentioned, where there are multiple genes involved, and then it could be different genes in different populations, or the same gene in different populations with different variants in those different populations. So
let's address that too. Yeah, let's address that, because so we know that one of the biggest issues in the genetic understanding that we currently have is that so much of the research was white, Caucasian, European descent, so let's dive deep into kind of the racial issues around that and what that really means for people if they are of color and they are trying to access the same kind of testing that I have. Does that mean that it's not accurate, their reports? I think that's probably the most widely asked questions, is like, does this mean that my report isn't accurate for me?
So that's a great question, and it depends on what type of test. If it's a carrier screening type of test, then the test itself is accurate in terms of the results of the test. However, if it's not testing a variant that's common in your population, then it's just not capturing that data, and it's just so there's some data and that's the importance of having diversity in research, because there can be a variant in one population that's not in another that causes the same problem. For like breast cancer, for example, that's the perfect example. So the BRCA mutations that you mention all the time, there's a whole world of other mutations in the BRCA gene that are in other populations that are referred to as variants of unknown significance, right? Gotcha. And that's partially due to the fact that most of the research was done on certain populations, but now that's beginning to change, and we are starting to identify other variants in other populations. But that's kind of the prototypical example of the importance of diversity in genetic research.
Yeah, absolutely. So important. So I just want to acknowledge someone that asked a question that's not really about genetics, but I did want to acknowledge her question. She has a partner with 12 million sperm count. And yes, you can successfully conceive. You might want to try. 12 million is enough. If he's got any other issues going on, slow swimmers or any morphology issues, we'd want you to connect with our urologist. We are doing a live. It was supposed to be yesterday, but he had an emergency surgery. Dr. Spitz is amazing. He's going to be on the show on March 12th, I believe, if that is the third Monday of March. I have to look at the calendar to make sure that's the right date. And also, if you ask that question in the app, then we can get that question answered for you from the app itself. We do have other urologists in the app. So I do think that March 12th is the date that he'll be doing a live, and you can come back and ask. But I don't think you should have any trouble based on 12 million sperm count. You can always try an ICI to get it even closer so that you have volume. But as long as they're swimming, then and you get the timing right, then you should be okay. April's been trying for 5 years, still has not gotten pregnant. And April, you're welcome, Jay. Have you done any genetic testing? I want to ask those who are still on with us, watching, has anyone done genetic testing? Have you done a 23andMe? Have you done carrier screening? Did you do PGT? We'd love to hear from you. What questions do you have? Are you currently pregnant? Are you considering NIPT? What does NIPT look at, Brad, that's different than PGT? And boy, we have a lot of acronyms in this field.
Right. NIPT is, I believe, referring to non-invasive prenatal testing. And that level of testing, again, are looking for gross anomalies that have been characterized, that are known, that you wouldn't necessarily if you had the choice to opt not to to carry a fetus with those, an embryo to term with those mutations because of the impact that they would have on the life of the child. And it's a delicate area. So it's a sensitive situation.
Right.
Parents with
Because you're
special
already pregnant.
needs.
Yeah.
Right, right, and so it's a tough situation,
Right.
and it's one where lots of hard decisions sometimes have to be made
Right.
around that testing when it's done.
But it's such a huge improvement doing NIPT over the old way of amniocentesis.
Right. The risks of amniocentesis to the fetus itself, although they were relatively low, they were still risks, and that it's a procedure that takes place that can potentially impact the pregnancy and the fetus, as opposed to a less invasive way that has significantly less chance of harming the fetus, but still get the information that you need to make your decisions.
Right. So if someone has been struggling to conceive, what is the sort of common genetic testing maybe? And if this is not your domain, you can tell us that we have to punt to like Dr. Chen. But what kind of genetic testing can people access if they are having trouble conceiving?
If they're having trouble conceiving, I mean, um, you can do some. I mean, carrier screening will give you information on potential genetic issues in your child once you get pregnant. But in terms of genetic, the infertility workup, it may involve some genetic testing, but it's usually more physiological testing, looking at ovulation, making sure that's occurring properly and regularly, and also looking at anatomical features, potentially an ultrasound, see if they're the fallopian tubes are blocked or something of that nature,
Right.
or some other type of anatomical issue. But in terms of having trouble conceiving, again, the genetic the research hasn't quite gotten there yet. We haven't done the studies on those types of populations. So we would
Sure.
need to look at, a large group of women and then look at them genetically, a comprehensive genetic look,
Sure.
and see what things are they have in common versus women who don't have these issues, to be able
Right.
to start to identify some of the genes that are playing
Sure.
a role in those.
And we know like there are genes impacting those with PCOS. We know that there's kind of up and coming markers people are looking at for endometriosis, right? And even fibroids. What else are you seeing in the research that's like we're starting to see some variants for at least infertility conditions for both men and women?
Right, those are the big three. Essentially, the PCOS is the most common ovulatory issue with regards to infertility, and then endometriosis is a close second, and fibroids are also very common, and so in terms of infertility in women, it can be broken down into an ovarian issue, a fallopian tube issue, or a uterine issue. And those are of course, the three organs
Right.
that are that make the magic happen, right? So,
Yes.
So that's how they are, largely categorized by those three in those three areas. But as you mentioned, those conditions, and then other than that, you have the chromosomal abnormalities that you can pick up on some of the screening that's done to like the NIPTs, et cetera.
April, oh my goodness, April's had all three. That's why she had to have surgery. Holy cow. That's a lot. I want to make sure that you've gotten proper emotional support for coping with all that, April, because you're not just talking about like trying to have a baby. It's also there are also conditions that impact you for your whole life, right? So these are not kind of isolated things that only matter when you're in your 30s trying to have a baby. So April, we are here for you. If, if we need to connect you with an expert or with an emotional support program, or even with a therapist, I just want to make sure that you've got a great team of support around you. And yes, I hope that that surgery that you did have is helping. I assume it's for probably endometriosis, if you did. And Rocky's asking, can ovarian cysts be hereditary? Is that something that, maybe we don't have the genetic testing for, but in general, is it thought to be hereditary?
Absolutely. So before there was genetics, the best indicator or the best proxy is family history, because
Right.
those individuals are related to you. And if they are affected by a condition, there is a chance that you may be as well. And if the more individuals in your family with a given condition, naturally and logically, the higher the risk for you as an individual, the more people you have in your family with a given condition. So yes, if you have a bunch of women who are very cystic in your family, then you can bet that your chances of being cystic are higher than the general population.
Got it. Okay. And then Jay, you've been trying for 6 years. So have you done an HSG? Do you know if your tubes are open? So to Brad's point, Jay, the ovaries have to work and they have to release an egg, a mature egg every month. The tubes have to be open. The uterus has to be receptive to an implantation. But for 6 years, and he has 12 million sperm count, I would want to make sure that you're fine. Now, he has plenty of sperm to work with. If you do need IVF, they would do IVF with ICSI probably to ensure fertilization, but check in with us and let us know what kind of workup that you've already done. And I think that would be helpful. And Brandy's asking, if you have a cyst on your right ovary, can you still get pregnant? Absolutely, Brandy, most cysts go away. So Dr. Chen always talks about this on our Monday motivation shows. She's an IVF doctor. And then every other Tuesday we have Dr. Sherry, and we just did an episode 2 days ago with Dr. Sherry that's in our IGTV if you want to check. We talk a lot about cysts. So most cysts go away on their own. Some need to be monitored and they can get big and they can burst, and that can be very painful. If you have recurrent cysts, if it's a single cyst, that happens sometimes just hormonally. But if you have recurrent cysts or they are painful, or, they do become problematic, then it's time to get, a specialist to check you out and see what's really going on, not just physiologically, but in blood work too, hormonally.
Yeah, I would add to that. It also depends on the type of cyst. Those simple cysts, as you mentioned, can resolve on their own, but something like a dermoid cyst is a little something different, and that would need to be, addressed surgically.
Haramex is asking a great genetic question. They did expanded genetic screening against 283 genes. Everything turned out great, all negative. However, the donor they chose for sperm was tested only against 17 genes. They were negative, but what's the potential risk if they looked at, the 17 that probably the sperm donor bank did were the 17 most prevalent, right? Expanded carrier screening goes to like that next tier down, which is less common. So should they be concerned, and should they ask for the expanded carrier screening on this donor they selected? Or because they're negative, will it not matter?
So this is the thing. The genetic tests will only give you information on what it tests. So
Right.
you have the information from the initial test, and if you're concerned, you could then opt to get another more extensive test, a more comprehensive test, and to look there. But again, we still don't know all of the genetic variants and the genes that are driving every single condition. So you can test. We can only test what we can test.
Right.
It sounds bad, but
I think we could break it down pretty specifically, because I had this exact issue with Dante. So Haramax, my son is donor conceived, and back 13 years ago, there wasn't any routine genetic screening going on in any donor, if you can imagine. There was no genetic screening being done on donors. I had no genetic information on him. I just had a spotless health history on paper. But I had cancer at the time. So of course I went for the donor that had a spotless health history. However, this donor had a family that reported a hearing loss condition in their child who needed a cochlear implant. So my donor was retired early, and they went back and did genetic testing on that donor and determined he's a carrier of this specific gene. That right when I was about to do my embryo transfer, they counseled me and said, if there's only one carrier, you have, X risk, meaning if I wasn't a carrier of the same, I would have, I think, a 1 in 4,000 chance of my child having, having the issue manifest itself. Whereas if we both were carriers, it was like a 1 in 400 chance. Still not like a crazy high prevalence, not like 50-50, right? And so I couldn't delay my embryo transfer. And I said, these are the only embryos I have. And since every single one of them might, have this issue, I'm not going to go and create the probe for PGD and for that issue. To me, that felt like it was a tolerable issue. And my son didn't have that hearing loss. In fact, he has like hypersensitive hearing. I feel like he can hear something 18 miles away. It's hysterical, but my son is a carrier. So the donor did pass on to my son the same variant. So now my son, when he goes to have a family, needs to have that conversation. So Brad, I guess my question to you is, of the 265-ish or 66 genes that they didn't test on the donor, is it going to be a similar scenario where most of the big genetic issues that we're concerned about require two variants, one from the X chromosome, one from the Y chromosome, for the risk to be super high?
Right. And so that is all dependent upon the mode of inheritance of a given condition or disease, for example. So what you're referring to is what we refer to as autosomal recessive genetic conditions, where there are two copies required for the phenotype or the condition to manifest.
Got it.
That's opposed to something that's autosomal dominant, where there's only one copy of a given gene required.
Okay.
So similarly, so my son, I passed on the sickle cell trait to my son, and when it's time for him to have children, he will need to be aware of whether or not his mate has trait as well, because if she does, then they will have a 1 in 4 chance of having a child with sickle cell, as opposed to something that's autosomal dominant, where only one copy is required for it to manifest.
So to put her mind at ease, she could work with a genetic counselor who can actually tell her, out of the 266, I think I'm doing the math correctly, out of the 266 genes that they didn't test the donor for, they could actually list out, hey, all of these are autosomal recessive, so you can put your mind at ease, everything's going to be fine.
Right, because you don't have a copy of that variant.
Right.
That would be required in an autosomal recessive type of
yes, got it. And if any of them turn out to be autosomal dominant, Haramex, just know that you're allowed to request expanded carrier screening on your donor. They might ask you to pay for it, but they do store those samples on the donor so that they could do expanded carrier screening, and you're allowed to ask for that. And if you need a referral to a great genetic counselor, we have amazing ones that work with us. Very cost effective. Brandy, I would keep testing for pregnancy. If you have been getting normal periods, even though you had that cyst on your right ovary, I would continue to test, because if you're like clockwork and regular, I would continue to test. April, you've gone through a lot. Are you working with a great integrative medicine, doctor on your case, or a naturopath like Dr. Raquel? She got hit with everything, like all the things that you can get, I feel like April's dealing with. So I just want to make sure that you have like a great team around you to get good support. And Jay is saying, no, the doctor won't help. She's saying she's okay, but I don't know because her youngest is 8. So Jay, if you're just with your OBGYN and she's saying you're okay, you need to find a different OBGYN. You're not okay. If you already have a sperm analysis from the male partner and he has 12 million sperm, then there's something going on. Most OBGYNs don't do an HSG to see if your fallopian tubes are open. So we advocate for getting in for a full workup with an IVF doctor, not because we think you need IVF, but because we think that you need a full diagnostic. There could be something very simple going on, like you have blocked tubes and didn't know, and that would explain everything. And then you can make a plan. But 6 years, it's not okay for your doctor to say she won't do anything because she says you're okay. You're not okay. And we're acknowledging we see you. And we know that, and we absolutely are here for you if you want us to set you up in your community with the right type of medical consult, if we need to find you a free consult, a reduced fee consult, if you need us to work in your insurance. Either register on the Fertility Answers app and let our fertility advocacy team help you by free text message. Slide into our DMs. I can help you right from Instagram, whatever you need, Jay. But I do advocate for you getting a different doctor. Because when it takes that long and you already have one child, everything's not okay. And Haramex, you're welcome on that information.
Is Jay the individual who said they had a cyst on their right ovary?
Jay's been trying for, let me go back up actually. Jay is not. She's been trying. She's the one whose partner has 12 million sperm, which isn't, in donors, we usually see 20 million, but still 12 million is probably not the cause, not after 6 years. One of those swimmers, a female swimmer, has endurance. She could have made it. If you have confirmation that you're ovulating, Jay, too, that could be another thing that you could do at home if you're not ready for a full consult. Uva Life is the product that we love to recommend. And because it measures progesterone, and if you release an egg every month, you'll have a progesterone increase. And if you get pregnant, you'll have a progesterone stay steady. And so that's something to just know too. You can always go to the Uva Life. I think you get 10% off if you put in Fertility Answers. And Brandy, we work with people who are on all kinds of insurance. So yes, Brandy's the one who has a cyst on her ovary. So don't worry about the type of insurance. We'll find somebody to work with you and work within your budget. So don't worry about that. We're free. We operate as your fertility advocate to help you get on the right path. Okay, so that was a great chat. I want to thank Avrio again. Follow Avrio, you guys. A lot of this type of genetic content, we are sending their way, they're sending our way. So we have a wonderful relationship with them to share content. It's just Avrio Genetics. That's their Instagram handle. AvrioGenetics.com is their URL. And before we wrap up, hi, Jessica. She's asking good supplement for her husband who got an abnormal sperm analysis. Go to Theralogics. They've got a great male prenatal. That's what we recommend, is going to Theralogics. They have a great one. And if for some reason you don't love it, DM me, Jessica, and I'll give you a couple other options. But we really love the brand Theralogics and what they have going on. Although I will tell you, Avrio is coming out with their own supplement line too. So stay tuned for their line as well. Hopefully they have a male prenatal in that too. All right. So thank you, Avrio, so much for bringing this kind of chat to the table. I know this is really dense information, you guys. So Brad, great job in breaking it down in a really simple, easy to digest way. I feel like we're getting like our own little mini accelerated PhD in genetics. It's kind of like doing those MBA weekends where you come out with your certificate. So every Thursday, you guys, is going to be Decoding Thursday. So we have 5 more of these. It's going to cover specific topics on genetics. We'll have a genetic counselor that Avrio is sponsoring. We'll have a patient who's going to go through epigenetic testing, the first of its kind ever in the world. And she's going to come on and share her experience to see how CBD has maybe helped her with other areas of her life, but maybe is negatively impacting her ability to conceive, and what she can do about it. So genetics is never a life sentence. There's always something you can positively do to optimize your own health and well-being. We love you, and we'll see you next time. Thanks, Brad.
Thanks. Take care.